In advanced molecular biology course, initially we are discussing the cell cycle, and in my previous lecture, I gave a brief summary of different steps in the cell cycle. Today, we will study some of these aspects in detail, and the first is the S phase, which is the DNA synthesis phase. DNA synthesis comprises of DNA replication, where two problems are for the cell. One is it must occur with extreme accuracy to maintain the next cell generation, and every nucleotide in the genome must be copied once and only once to prevent the damaging effect of the gene amplification. The cell cycle, the interphase, which is the first phase, it comprises about 90% of the cycle, and the G1 phase, which is a growth phase, then S phase, which is the synthesis of DNA phase, and G2 phase, that is the preparation for the cell division. Then starts the mitosis or mitotic phase, and in the mitosis, it is the cell division, and then cytokinesis, that is the cytoplasm division. Now, S phase, that is the synthesis of DNA phase, it comprises of the synthesis phase, which is the essential aspect of the cell cycle. During S phase, a number of events additional to chromosome replication, they also take place. And initiation and completion of DNA replication defines the beginning and ending of the S phase of the cell cycle. Now, DNA replication begins at origin of replication, which are scattered at numerous locations in every chromosome. During S phase, DNA replication is initiated at these origins when a DNA helicase, an enzyme which unwinds the double helix, and DNA replication enzymes are loaded into the two single-stranded templates. Initiation phase of DNA replication, the first step there is the early G1 inactive DNA helicases and pre-replicative complex of pre-RC, that is the licensing of replication origins, is formed. Origin recognition complex ORC, a multiplayer of protein, binds to replication origin throughout the cell cycle. Although the onset of S phase, chromosome duplication is triggered by the activation of SCDK, that is the cyclone-dependent kinase. SCDK activates the specific initiator proteins by phosphorylation that unwinds the DNA and initiates its replication at the replication origin. Remember, once replication origin is activated, SCDK also inhibits the proteins that are required to allow that origin to initiate DNA replication again, as is shown in this cartoon. The different initiation, elongation, and MCDK activation and chromosome segregation steps, they are shown. In summary, what we have discussed today is that the duplication of chromosome in S phase involves accurate replication, duplication of chromatin proteins, and the chromosome duplication is triggered by the activation of SCDK, which activates the protein. And once a replication origin is activated, SCDK also inhibits proteins which allow the origin to initiate DNA replication again. Another, in nutshell, is that each origin is fired once and only once in each synthesis phase, and it cannot be reused until the next cell cycle.